I'm very excited to be here and have the opportunity to present the first results of the RNA-Atlas, which is a very much ongoing work. There have been several consortium efforts to characterize the expressed elements in the human genome. These that I show here are three well-known examples, and all of them have been of great importance to expand and improve our picture of the human transcriptome. However, this transcriptome is still largely incomplete, and this is partly due to the limitations in the extent of sequencing methods employed by these studies. For instance, all of them focus mainly in polyA-selected libraries, while only some of them include complementary library preparations like small RNA, and most of them include only unstranded data. So to fill in these missing gaps, we settled the RNA-Atlas, in which we included a comprehensive sample cohort consisting of 45 tissues, 160 normal cell types, and 93 cancer cell lines. And for each of these samples, we generated three complementary library preparations: small RNA, polyA, and total RNA libraries, all of them stranded. And we sequenced them at high coverage with Illumina technology, yielding a total of 65 billion reads that virtually contain information on all RNA molecules present in the human transcriptome, including polyadenylated and non-polyadenylated RNAs, circular RNAs, and microRNAs, and across a very heterogeneous sample population. So this is a very simplified overview of the workflow we employed to generate this transcriptome. I don't have time to go into detail here, but basically we applied specific tools for the different types of data, and we then integrated specific reference databases to identify known and novel RNAs from the biotypes of interest. There are large linear RNAs, circular RNAs, and microRNAs. And we also applied a first expression filtering to filter out unreliable, lowly expressed transcription. And the generated transcriptome contains most of the RNAs that were annotated in the reference databases previously. We retrieved 97% of the genes annotated in Ensemble under the biotypes protein coding, long intergenic non-coding RNA, and antisense. And although a bit lower, we also identify most of most of the circular RNAs that were annotated in circ-base, and most of the major microRNAs that were annotated in mir-base. But most importantly, we also found thousands of potentially novel RNAs. 60% of all identified genes are potentially novel genes. And zooming in into the different biotypes, here I show the novel genes in lighter colors. So we see that the vast majority of these novel genes come from the non-coding portion of the transcriptome. We only identified 250 potential novel protein coding genes, while the majority of microRNAs, circular RNAs, and long intergenic non-coding RNAs are novel, with the fraction of long non-coding RNAs being as high as 3/4 of all link RNAs. So the question is, what are all those novel link RNAs? And well, a striking observation is that the wide majority of these are actually monoxonic transcripts. And here I show an example of one of those novel monoxonic link RNAs that has high read coverage across many samples. And we saw many examples like this one But of course, the fact that, that these monexonic genes have high expression, high read coverage, doesn't necessarily mean that these are functional transcription. And indeed, most transcriptome assemblies remove these monexonic things straight away. But we decided instead to keep them and to try to further filter them and try to validate at least part of them, reasoning that part of these link RNAs could be indeed functional transcription. So a way of filtering and refining our gene models and defining a stringent set is by integrated independent omics data sets from external cohorts. So we integrated evidence at two levels. At the level of RNA, we integrated cache-seq sequencing data from Phantom 5. And at DNA level, we integrated the chromatin states defined by the Roadmap Epigenomics Consortium. These are combinations of chromatin marks across the genome. And we took the chromatin states indicative for active transcription start sites and chromatin states for indicative of active enhancer. And for each case, we retrieved the closest peaks to our defined transcription start sites. And we consider a peak as evidence if it is within 500 nucleotides from our transcription start site. And this is how it looks like for known genes. Here I show for each RNA biotype and further split it in monexonic and multi-exonic, the fractions of genes with evidence. So in yellow I show the genes that have DNA evidence only. In orange I show the fractions that have RNA evidence only, and in green the fraction that have both DNA and RNA evidence. And as expected, we see that for protein coding genes, the majority of genes have evidence, while the fractions for non-coding RNAs are a bit lower. And for novel genes, the situation is similar. We see that the largest fractions with evidence are among the protein coding genes, and for non-coding RNAs, the evidence is lower, especially for the monexonic novel link RNAs, in which 40% of the genes have no evidence. But still, we observe that overall, 70% of all genes have either of these levels of evidence and constitute therefore the stringent set we defined and which we did the following analysis. Something very interesting of having matching poly-A and total RNA sequencing data is that we can use this information to annotate the polyadenylation status of our genes. So if we take sets of known genes, of known polyadenylated and non-polyadenylated genes, and we compare this, their expression in between poly-A libraries and total RNA libraries, we see a clear separation of those two sets, with the non-polyadenylated genes much more highly expressed in total RNA libraries. So we can then use the ratio of expression between poly-A and total RNA libraries to define a cutoff to separate polyadenylated and non-polyadenylated RNAs based on this on the reference known genes. So we defined this cutoff and we classified our genes, and we found that the majority of protein coding genes are actually polyadenylated, and we also see that most antisense transcripts are polyadenylated. But for link RNAs, the situation is the opposite. We see 60% of these genes are non-polyadenylated, and zooming in into this biotype, we see that the vast majority of these non-polyadenylated genes are actually monexonic link RNAs. Anyway, we also found a fraction of multi-exonic link RNAs that are non-polyadenylated, and this highlights the importance of using total RNA sequencing to profile a part of the transcriptome, of the non-coding transcriptome that is non-polyadenylated. And so far, I have shown you how we generated our transcriptome and different approaches we took to further filter it and annotate it. And in the last part of my presentation, I would like to focus on a different type of analysis we have been developing that is related with assessing the biological signal contained in the different RNA fractions. And one way of doing that is through computational deconvolution of tissue expression profiles based on the expression of cell type specific biomarkers. And this work has been performed by Francisco Avila Cobos. He's another PhD student in our group, and he's presenting this work this evening in poster number 5, so I really encourage you to go and visit his poster to get more information about this. So, but I will briefly explain it. So the idea is that we can select cell type specific biomarkers from the different RNA fractions, and then we can evaluate the expression of those biomarkers across tissues, and then through computational deconvolution, we can get the proportions of those cell types in each tissue, and then we define, for a given tissue, we define the signal as the sum of the proportions of the cell types that are components of that tissue. So for instance, in this small example, the signal here for liver would be the sum of the proportions for hepatic stellate cells and for hepatocytes, that are the cell types that form part of that tissue. So we perform this deconvolution for all RNA biotypes separately. And so for each tissue that could be deconvolved, we computed this signal. And so if we compare mRNAs and lincRNAs, give a very similar distribution. In this plot, each dot is a tissue that was able to be deconvolved with the specific RNA fraction. And so we see that with mRNAs and lincRNAs, we were able to deconvolve many tissues with variable performance or signal. And for circular RNAs, we see that we were able only to deconvolve few tissues, but for 2 of them, the performance or the signal retrieved is very high. And finally, we wanted to do this analysis as a way of validating the before-mentioned monoxonic lincRNAs. So we did the convolution using only this specific fraction of the transcriptome, and we observed that we were able to deconvolve some tissues with both known and novel monoxonic lincRNAs, meaning that even in these monoxonic transcripts, there is some biological signal which could indicate that they have actually a biological meaning. So my take-home message would be that by combining complementary RNA sequencing methods and across a comprehensive sample cohort, we were able to generate a unique resource of the human transcriptome that provides a nucleotide resolution of all RNA molecules in the human transcriptome. And I didn't have time to show all of what we have done, but there have been a number of several additional analyses we have been performing, so I want to mention some of them. One is related with the assessment of variability in polyA-status across samples. We have observed that some genes show variable polyadenylation across samples, and we are now investigating that in more detail. We have been using intronic reads to get insights into regulatory mechanisms of non-coding RNAs over their targets. And we have performed imprinting analysis, and we are now working on alternative splicing analysis and tissues, cell type specificity of alternative splicing. And if you want to get more insights on some of these analyses, you can visit my poster, number 11, also this evening. And to end, I want to acknowledge all the people that form part of the RNA-Atlas consortium. We are very grateful to Illumina to sponsor this project. A special thanks to Gary Schroth and Steve Gross. And then from Baylor College of Medicine, I want to thank the lab of Pavel Sumasin, from Aarhus University, the lab of George and Kims, the group of Ashwin Unikrishnan from the University of South Wales, Sydney, and finally from my university, Ghent University, I want to thank the labs of Ivan Saiz and Tim de Meyer, and my lab, the lab of Jovan de Sompele and Peter Mestag. And thank you all for your attention.
That's a great resource. For the single exonic transcripts, do you see any particular cell types and tissue types where they are more abundant or enriched?
We haven't done this analysis yet, but yeah, we have seen some tissue-specific monoexonic RNAs, but yeah, we still are working on that, but yeah, it's interesting.
Are they more conserved?
No, I think in general they are not more conserved.
Our experience says mostly they are tissue-specific and they are very poorly conserved. Only 25% of them are actually present across species. So
okay. Yes, probably it's the case.
So I have two questions. The first one would be that it's been known for 40 years that some protein-coding messenger RNAs are not polyadenylated. And it occurs to me that many of them may be undiscovered because people always look at poly-A. Have you discovered conserved protein coding poly-A minus RNAs?
No, we haven't analyzed that yet also, but would be, yeah, also really interesting to see.
And the next question is,
yeah.
is the interest is whether the mono-exonic non-polyadenylated RNAs are also capped, and I guess the data you have would be from the cage-seq. So do you have the same degree of cage-seq support for those RNAs?
Can you repeat the last part of the question?
Do you have the same degree of cage-seq support for the mono-exonic non-polyadenylated RNAs as you do for the polyadenylated RNAs? Link RNAs.
Link RNAs. Well, actually, yeah, we have more non-polyadenylated monoexonic link RNAs with cage, but in general we have more monoexonic in that fraction. So, so yeah, we didn't analyze it. Yeah.
Have you seen any?
We haven't. That is also ongoing, but we have only compared with ensemble so far. But yeah, we actually it's the next thing to do. Yeah.